I'm Mishra from the Electrical and Computer Engineering Department at ISR, and we are here today for the second in the series of all ISR Distinguished Walkout. And it's a great pleasure to have with us today Professor Mishra from New York University. So I'm going to tell you a little bit about Professor Mishra before we start his talk. Professor Mishra received his PhD from Carnegie University in 1985 after finishing his earlier science and engineering education in India. He has been since then on the faculty of the Courant Institute of Mathematical Sciences at NYU. He currently holds an appointment as Professor of Computer Science and Mathematics at the Courant Institute. And currently, he also holds professorships in cell biology at the School of Medicine at NYU, as well as an adjunct professorship of the Courant Institute of Fundamental Research in Bombay, India. So, as a personal remark, I met Professor Mishra in 1991 during the symposium as part of the American Association for Advancement of Science annual meeting here in Washington, D.C. It's about almost 14 years ago. And at that time, he was working on robotics and computer science mainstream problems. And he spoke about grasp problems, robotic mechanical hands, and the interesting idea of pentadactylity, or five-fingeredness. Why do we have 5 fingers? Or can we do with less of it? Or do we need more for certain purposes? So, it was an artful combination in my memory of convex geometry, statics of non-frictional contact by means of fingertips. It had some wide-ranging intellectual historical remarks, some of which found their way into a later paper he wrote, in which the remarks go from paleontology to modern technical stuff. And the talk itself was quite interesting, I'm sure I remember. And this is in particular the hallmark of his communication style and his scientific approach. It's broad, deep, and engaging at all levels. In the intervening years, we were running into each other, and I learned about his growing interest in matters biological, including bioinformatics, and the technology for observing, modeling, and investigating biological systems on cellular and subcellular scales. He approached the questions with energy and insight. His contributions in this area are recognized by the patents that his group has garnered, the interest of venture capitalists in his ideas, the creation of companies, as well as publications and invitations to lecture on synthetic systems biology at prominent venues. In his recent work, he's focused on mathematically well-founded software technologies for exploring the cellular realm. So that's the subject of this talk, Telecom.
Thank you. So I'm not going to go too deep into all the sciences, so I'm going to just tell stories. And thank you, Krishna, for the great introduction. Very flattering. So I'm going to start my story with this character. So one of my favorite characters. I don't know how many of you know who he is. He's an interesting character for us. He's Robert Hooke, who lived between 1635 and 1703. And the reason he is an interesting character for us, among many things, is that he was the first one to observe a cell. So Robert Hooke was an experimental scientist, a mathematician, and an architect. Actually, with Christopher Wren and John Evelyn, he redesigned London after the Great Fire of London in 1660. These are the years with fire and plague, and so wonderful to see what that can do to scientists. And he was considered the English Da Vinci. He was so broad in his interest. But the reason he is interesting for us is because of his work on micrographics. He was the first one to observe cells, and they actually look like cells of monks in a monastery. That's why they call them cells. And also another interesting thing was when Newton wrote Book 2, which later became Wikipedia, he referred to Hooke as Clarissimus Hookeus. Except in the later versions of the book, Hookeus disappeared completely, as if he never existed. And the reason for that was Hooke became involved in a dispute with Isaac Newton over the priority of the discovery of the inverse square law of gravitation. It was a bitter dispute, and so there's an interesting story to tell. So Hooke wrote to Halley, who was the secretary of the Royal Society. He said, Haydn's preface to Principia is concerning those properties of gravity which I myself first discovered and showed to the society in the years since which, of which Mr. Newton has done me the favor to print and publish as his own inventions. Not very happy, of course not happy. And Newton was not happy either. So he wrote back to Halley saying, now is this not very fine? Mathematicians that find out, settle, and do all the business must contain themselves with being nothing but dry calculators and drudges, and another that does nothing but pretend and grasp at all things must carry out all the inventions. I believe you would think him a man of strange, unsociable temper. But Newton actually went one step further. He actually wrote a letter directly to Hooke, a very famous letter. And there is a sentence there, if I've seen farther, he meant farther, than other men, it is because I've stood on the shoulders of giants, that's Galileo and Kepler, and you, my dear Hooke, have not. We often quote this statement, but we forget the context and what he had to say about Hooke. And then, of course, because of this dispute, there was a need for settling that. So they invited Christopher Wren, and Christopher Wren, in his famous letter, said there's a great distance between a glimpsed truth and a demonstrated truth. So, in some sense, we're still fighting that debate about the truth. And so to some degree, I'm going to talk about that and why we're still fighting the fight that Hooke and Newton started, and do we have better tools? So let's start with micrography and photography. So here is actually micrography. So these are the cells that Hooke saw, and these are beautiful pictures of micrography. And in there he also describes what he meant by the truth. The truth in the science of nature has already been too long made only a work of the brain and the fancy. It is now high time that we should return to the plainness and soundness of observations and material and obvious things. So, similar to the kind of ideas that Bacon had been talking about, and also to some degree Leibniz and others. So, in his view, biology, the science of this kind, had to be purely an observational science. The truth has a basis only if you can observe. You can make hypotheses, you can go do an experiment, you can observe, which will either strengthen your hypothesis or refute, and that's the nature of the truth. And here is Principia. And it's an interesting thing. There, Newton actually wrote down this motto, hypothesis non finio, I frame no hypothesis. So what was his vision of truth? Much better than Newton, Halley actually writes about that. He says, truth being uniform and always the same. It is admirable to observe how easily we are able to make out very abstruse and difficult matters when one's true and genuine principles are obtained. So it's driven a common principle that will explain many different things. So the debate between the proximate truth and the ultimate truth. And this is sort of from Halley's article, The Truth Theory of the Tides, extracted from that admired treatise of Mr. Isaac Newton entitled Philosophy Naturalis Principia. And I love titles like that. We don't try to do this. So in that context, it'll be interesting to look at yet another story. Again, a story from biology. It's a very sort of story that's at the core of developmental biology, but also touches computer science. So the story begins with Alan Turing. Of course, we all know of Turing, because Turing machines, Turing test of intelligence, and a number of other things. But Turing also proposed a basis for morphogenesis, a chemical basis for morphogenesis. His interest was because he wanted to understand how human brains develop from a single cell and could do these complex calculations. And it's a paper that came out in 1952. Actually, Turing is fairly well known among biologists for his work. An interesting career. So one way to go about it is to look at his model of reaction and diffusion. And it's sort of in the tradition of Newton, in the sense he's looking for principles. He had no experimental basis for the kinds of models he proposed. So he wanted a very general program for modeling embryogenesis. The model is a simplification and an idealization, and consequently a falsification. He admits that there is no basis to the principles he is proposing. In that, he introduces the notion of morphogen. Now, morphogen means something much more concrete, but in his paper, it meant simply the kind of substance, it's an abstract object, kind of substance concerned in this theory. In fact, anything that diffuses into the tissue and somehow persuades it to develop along different lines from those which could have been followed in its absence will qualify as morphogens. So you have some object or morphogen, and you can write down the equations of how the concentration of morphogen changes over time and space. So there is temporal derivative. So the rate of change of the concentration, and it diffuses with some diffusion constant. So there is a second spatial derivative and a first temporal derivative. And to this, you can make it more interesting if you also introduce a chemical reaction and consider two morphogens. Now there are two morphogens, A and B. They react with each other. A has a diffusion constant as B sub A, and B has a diffusion constant B sub B. They're substantially different from each other, and A and B interact chemically. So A's concentration changes as a function of A's own concentration, as well as B's. And similarly, B's concentration changes as A. In fact, these are nonlinear, but very mildly nonlinear. In fact, both these functions are affine in A and B. So degree 1 in A, degree 1 in B, and you can write this thing. So this is the classical reaction diffusion equation, and you can actually take the reaction diffusion equation of your choice and program it into a chemical reaction fairly easily. So here is a scheme for taking this reaction and constructing a reaction diffusion equation. You have to remember that when Turing did all of this, he only had a Turing machine. He really didn't have much of a computer, so a lot of the patterns, a lot of the asymptotic solutions that he talked about were only things that he could write down analytically. But subsequently, people have done computer simulation, and you get patterns like this. And the interesting thing is that you get sort of spots and stripes, things that look very biological. So one could propose that most of the pattern formation in biology is governed by reaction diffusion equations. And the question then becomes, what are these morphogens? And Turing actually thought of these morphogens as proteins, and these are metabolic reactions. The question was, what is the role of genes? And he really didn't think that genes are very important. He thought they are simply catalytic, and they only influence the rate of reactions. So if you're genetically different from somebody else, it's because you're doing the same chemical reactions, but maybe you're synthesizing at a slightly different rate. So he did say, unless one is interested in comparison of organisms, these things, genes, may be eliminated from the discussion. So genes, in this theory, has no really any interesting role in developmental biology. I'm sure a lot of biologists are saying it's not true. And this is not true. And we found in the next year, 1953, Watson 3 came up with a double helix. They did not necessarily describe the role of genes in developmental biology, but they did understand that it played a much stronger role, and the famous line is, it has not escaped our notice that the specific variant we have postulated immediately suggests a possible copying mechanism for the genetic material. And probably they didn't even think of something that's much more different from Turing's. And that's sort of revealing when you think about Crick's central dogma. So let's do a little bit of a tour of genomics. So for our purpose, we'll think of a DNA molecule as a long sequence of nucleotides or bases. So there are A, G, C, Gs. And some regions of these long sequences will play a role in transcription. They'll be transcribed into mRNA, and then will be translated into proteins, and that will give you all the parts, the part lists of biology, and then they will interact in some complicated way in trillions of cells to make you and everybody else in that, all the living organisms we know. And the central part of this thing is a dogma that says there is information flow in one direction. There's DNA. DNA gets transcribed to RNA. RNA gets translated into proteins. And these proteins are going to take part in metabolic reactions to do interesting things. You change the DNA, and you will change RNA and then the protein. So presumably, you'll change these rates. You will change the reactions, but only changing the rate of reactions. It's also very Darwinian, because here you'll change the genotype, and here you'll change the phenotype. So the information flow is always in this direction. So Turing was sort of right to pick this. Right. But it turns out that this picture is not correct. The reason is that some of the proteins come back and control the rate of transcription for what gets transcribed. You can turn there is a strong regulatory role that these guys will play through transcriptional factors that will come to promoter sequences that are upstream of the gene and can turn it on and off. That sort of changes the whole picture. And now you can postulate that these genes may play a role in developmental biology and pattern information. So, just a little digression. While all this was going on, what was in biology interesting in mathematics? So it's just an interesting quote from Eric Ponder, who was then director of Long Island Biology Association, which became Coastal Harbor Lab. And there he said it is futile to conjure in the imagination a system of differential equations for the purpose of accounting for facts which are not only very complex but largely unknown. The kind of things Turing was doing was not really very useful. There's no point in biologists to waste more time on this. And in fact, somebody who succeeded Eric Ponder, E.B. Wilson, in fact wrote down 5 axioms, which he called axiom of latitudes. And that describes why mathematics is not going to play a role in biology. So the first one is science need not be mathematical. So, and simply because the subject is mathematical, it need not have the scientific empirical curve-fitting. Actually, we've said change from this to data mining. Maybe without other than classification of significance, growth of an individual should not be confused with the growth of an aggregate of individuals. Different aspects of individuals or average may have different types of growth curves. So, if you accepted these axioms, which are self-evident truth to biologists, then of course mathematicians need not apply. So that was. So the story should have ended there. Turing did something that was wrong, and so. But something interesting happened. So this is the next part of my story. People actually very carefully started looking at pattern information in Drosophila. So they were interested in segmentation. So in particular, they wanted to see the pattern formation in the body plan, anterior, posterior patterns, and dorsal ventral patterns, as well as along the germ layers, so the ectoderm, mesoderm, and endoderm. And most of the theories can be explained by a very simple principle, and that's the French flag model, or more formally called positional information. In this picture, you have a set of cells. So there are 6 cells. And there is some chemical. Now, morphogen is some chemical here, for example, could be macronal mRNA, could be other things. And it has a gradient. And each cell can find the gradient and compare it to the threshold. And depending on its value relative to the threshold, will do various things. So, for example, here is a gradient, and if these cells sense a gradient is above the upper threshold, they will turn blue. If it is below the lower threshold, they will turn red, and if it is between those 2 thresholds, they will turn white. And the interesting thing is that if I put more cells, I will still get a blue, white, red French flag pattern. Okay, so we can make French flags by simply extending this. American flag is more difficult, but synthetic biology could be useful. Okay, so how does that work? So here, we start with the flag A. So there's a wild-type A. There is some macronal mRNA. There's a bicoloid mRNA. They're concentrated on one end, and there's a gradient. And wherever there's this high concentration, you get a head. How do you know that? If you take away bicoloid, you get a larva that doesn't have a head. Mutant larva with that head, and you put it in the middle, and get a head in the middle. So you can actually move your head in the larva just by changing this gradient. Okay. So it's purely experimental work. So then what happens next? You can now look at a set of genes called gap genes, and then there are another set of genes called pair rule genes, and they'll cascade one after another, making these waves of gradients. Right? And they'll turn things on and off, and it's all based on this same positional information theory. And at the end of it, you'll get a flag. That's sort of a very short description of a long story. Okay, so here the main players are genes, and they are controlled turning on and off. It's a programmed pattern information. It has nothing to do with kinds of pictures Turing was proposing. And of course, this was a big thing, and there was a Nobel Prize to Edward Lewis, Christian Russell-Involler, and Eric Bushhaus that year. And they were purely by doing sort of this mutation in an archive experiment, they created this story, the picture of how things are programmed. Okay. So now what you can do is turn it into a circuit diagram. So to sort of understand what the circuit diagram means, remember we had DNA, the genes, they got transcribed into these mRNAs, and these mRNAs got translated into proteins, and some of the proteins come back as transcriptional activators and turn other genes. So you can draw this as a picture. So here is a hedgehog mRNA, that's making a protein, hedgehog protein. So the RNA will be little letters and protein will be capital letters, and hedgehog itself is turned on by EN, and so on and so forth. Okay, so once you write this down, you can actually write down the differential equations again for each of the transcription, which will be some sort of Michaelis-Menten type or field type equations. So it will be a first order differential equation with a rational function on the right hand side, and the protein translation is much simpler. It will be linear ODE, which will depend on the concentration of the mRNA and the degradation rate of the protein. Okay? So in fact, we'll see its ODE to look like first order rate equals a rational function that tells how things get transcribed, so something that describes synthesis minus something that degrades. So even if this looks complicated, you can write this down. But interestingly, I don't think you can see very well, you'll see that this will not work. And there are a few other reactions that you need to introduce. One is this, and the other one is right here. These two reactions were not in the picture that the biologists had proposed. And without that, they're going to make a couple of oscillators and there's no pattern. Okay, so Gary Odell and his students, mostly undergraduates, actually wrote down these reactions and did simulations and found that this picture that they had created never worked. And then when they introduced those two reactions, so we incorporated these two remedies first. With these lines, with these links installed, there are many parameter sets that enable the model to reproduce the target behavior, so many that they can be found easily with a random sampler. That means you can just randomly put rays of reactions, and still you will get a plot. So experiment resulted in something, but that was not a complete picture, there was something completeness. And here is how you found it, and these are the rates of reactions, and these are the ranges from which they did random sampling. And these are the reactions. They look ugly, but you can, so this is wingless mRNA. It's being transcribed, the transcriptional factor is CN, and there's some bucket, I think CL, and there's being repressed by CN. So you get this rational functions, and you have some degree, these are the field coefficients, and there are some parameters. And then also modulated by IW, and then there is WGR, so this is the degradation rate. So these are all normalized by rewriting things, and then this is the protein, rate of change of proteins, and that will depend on the mRNA and other degradation processes. So they look ugly, but they're actually fairly simple first order proteins. Again, there are something like 14 of them.
So this is very solidly deterministic in character.
This is solidly deterministic. This is a kinetic mass action. So you're assuming the copy numbers are so high that some of the stochastic effects are not important. But right now, people are actually gotten very interested in the stochastic parts because there's a lot of interest in the noise level in the decoy, and the idea that these things are very robust, even if I change the concentration of original decoy, all those things have become very interesting studies. And people are building models of those various kinds of noises and transcriptions and things like that. But this is a deterministic model.
How about the delay?
How about
the delay? Seems instantaneous to the delay.
These seem instantaneous because I'm just writing down just the interaction between these things. But what will happen is each genes will get turned on one after another. And you can write that down in terms of delay equations. But that will make it a little bit more complex. And we'll come back to that, why we need to do a little bit more than just writing down this ODEs. So now the question is our final answer. Do we now understand pattern information and why? And the idea is that if you look at biology, biological truth, you do have to worry about these issues. There are cognitive flaws, things we didn't understand, that qualitative simplifications, and we make assumptions that are extremely general. So we need some sort of process that allows for reasoning, simulation, model building, and experimentation all to happen together in order for us to get to what we would like to call the biological truth. Okay. And here are some examples. My favorite examples are the ones field hypothesis. There are just two Nobel Prizes for this. And then last year, people actually did experiments where they could synthesize these things and observe. But there are a lot of things in this model that are incomplete. Cell cycle machinery. There are good yeast cell cycle models. Mammalian cell cycle is another story. There are two contradictory theories for muscle contractility. Both theories are widely accepted, almost in equal footing. Various processes involving cancer. We believe the war against cancer has been won in the sense we understand what causes cancer, but if you look at each component, cell cycle regulation, angiogenesis, DNA repair, apoptosis, cellular senescence, tissue space modeling, all these things still remain poorly understood. So what do we do? Pictures and things. Systems biology. So what is systems biology? It's sort of an amalgamation of many fields. So this is my favorite explanation of what systems biology is. This is system biology, coloring book, it's a good business style. Interestingly, the names are related. Interpretive biology, integrative biology, computational biology, bioinformatics. And it sits between two interesting fields in my picture. And I define it as a field aspiring to combine the mathematical rigor of numerology with the predictive power of astrology. So we want something that's mathematically very rigorous. So it looks like physics, and yet should be able to predict everything. Whether you'll get a cancer or some genetic disease, or if you knock out this gene in the fly, you'll become a super fly, things like that.
How about the rigors of the work?
Yes.
Understanding exactly how to come to the other world. Those system means
Right. 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 This is not to be taken too seriously. Just I probably, in my secretly, I agree with you. But this is right. But this is sort of why it is interesting. We do really want to do something that's mathematically rigorous and has interesting predictive laws. So in that context, I'm going to talk a little bit about what we do, computational systems biology, which is more in the direction that you talked about. And this is, I should tell you that this is part of a large DARPA funding BioSpice, which tries to emulate the SPICE project in VLSI. So wants to create similar tools for biologists. And given the success they had, DARPA had in SPICE, they want to do the same sort of thing here. So the idea is that by combining already existing tools and theories, so that we know in dynamical systems, kinetic analysis, computational theory, and logic, we should be able to create a set of tools that could be very useful for the biologists. And one of the things that I am trying to emphasize, the goal is not to simulate biology, because I really don't get any understanding, even if I can simulate every mRNA, every protein, how they fold, how they degrade, but would like to simulate the reasoning processes that biologists use in thinking about large complex systems, in designing experiments, in doing the kind of things they already do, but at a much larger scale. I mean, that's actually necessary. I mean, this is the story that if you look at something like a gene like p53, there are at least 8,000 papers. And there's not a single p53 expert who has read 8,000 papers. So what happens, you write a paper and you look for a reviewer. So clearly, the complexity has become far more difficult than what can be handled at this point. Okay, so I'm going to start with very simple sort of ideas, very sort of engineering ideas. So I'm going to start creating a modular structure out of these simple reactions. So I could have an irreversible reaction. So X1 goes to X2. Think of these as species, some chemicals. Or I can have reversible reactions. And the rates are different in either direction. Or I can have splitting reactions where something degrades into its components. Or I could have multiporization or synthesis, something gets synthesized. I could have similar reactions, except I may have some stoichiometric constraints. Okay, so I will write it as these equations. And then I will have irreversible reactions modulated by some enzyme or modulated by some enzyme coenzyme. And so these are very, very important in biology. Because without that, every chemical reaction will be a futile reaction. Nothing interesting will happen. This is what makes things irreversible. And most of it is done by hydrolysis, where you consume ATP, most of the energy, to keep this reaction irreversible. So if you look at any biochemical reactions, you'll see a lot of interesting multimerization, phosphorylations, regulation, but in between you'll see these irreversible reactions, making sure the processes go in one direction, at least in one compartment. You can find a reversible, exactly the opposite reaction in another compartment or in another cell. The interesting thing is that all of these can be written as ODEs, and then they can be all composed to create bigger systems, and then those bigger systems can be composed to make hierarchically more and more complex systems. But independent of that, there is a way you can write them down in a canonical form. So you'll have, by introducing other variables, you can write down the rate of change of some species as a binomial, some monomial that describes the synthesis, and some monomial that roughly describes the degradation, and you'll have some complex algebraic constraint. So there's an algebraic manifold, and this is going to evolve in that. The reason I'm sort of interested in this is that at some point, if you want to use differential algebra to reason about this, there's a possibility that these systems could be proven to be isobaric. And if that is the case, a lot of progress that can be made in this area. But right now, we don't understand enough about differential algebra. And we don't have anything like Hilbert's basis theorem. We have a read-random Bush, but that's for radical ideas. But if it is isobaric, there are decision procedures, similar to Grobner basis and standard basis. That's a totally different story, but this is an interesting connection. So having canonical forms like this for biological systems gives you hope that you can do more than you could have done in other nonlinear systems.
What does isobaric mean?
Isobaric is a notion similar to homogeneous polynomials. So in homogeneous polynomial, you make all the degrees equal. Right? Here, what you do is you introduce a weight. This makes it isobaric. And you have different weights for degrees and the orders. And each term in a differential algebraic formulation should have equal weight. And once you have that, you could do something like a standard basis. Actually, if you look at Grobner basis, most of the interesting work happens in homogeneous case. And then you go back to affine cases. You could do the same trick here.
And what's the physical basis for those kind of constraint equations?
These are many of them are conservation equations.
Bookkeeping.
Yeah, it's bookkeeping. The interesting thing for doing this, instead of just writing down the equations and doing numerical integration, So there are errors you will introduce in numerical integration. So here, if you want to do exact analysis, it's better to do it here. We'll come back to this question. But we don't have a decision procedure. Actually, the membership problem is not known to be decidable or undecidable. That's the classical Gibbs problem. It remains open for like 50 years or so. Okay, so that's sort of it. So here is how you can write down glycolysis.
Well, why do you go get a name out without
In what sense?
I mean, I'm expected that you were going to taking anyway.
Okay, we'll introduce logic, but this is the most simplest case where I don't have any discrete interactions. We'll come back to that. In fact, we'll abandon the difference algebraic approach completely. And at that point, it makes it easier to just introduce more things. And the nice thing is that we'll see there that we'll have decision procedures. And we'll have assimilation results and stuff like that. So here is a classical glycolysis process. That's how you convert glucose into fructose, that converts to glycogen. And the interesting thing is that you can write down this as a hierarchical composition of the basic building blocks, which are basic reactions. And here's an interesting example. This is not a real biological circuit. It's a synthetic, an artificial circuit. Somebody took 3 genes, theta, lacti, and lambda CI, and pulled them together in a plasma. So the idea here is that the gene A, gene B, and gene C, each one has the promoter regions, PB, PA, and BC, and gene product makes ultimately a protein A, and it binds to PA, and it represses gene B. And gene B makes similarly another protein that binds to this, and represses gene C, etc. So it's like 3 NOT gates tied end to end. What does it do? It's a clock. It oscillates. And it has a time period that's the sum of the half-life of the protein degradation. Okay? So now you can take a plasmid like this and introduce it into E. coli. And you can also create another plasmid that has a GFP, which is a green fluorescent protein. They occur naturally, and they will glow. And the green fluorescent protein is going to be controlled by, I think it's state R, it's hard to read. And so what will happen is that as this thing oscillates, this thing will start blinking. So you have now equal blinking E. coli. And the nice thing is that the period of this clock is much larger than the division time for E. coli. So you'll see this colony growing, and they'll be blinking in lockstep. It's a nice display device of the future, E. coli displays. And so there are a lot of things one can do like this. And there's a big community of synthetic biologists who try to design circuits and introduce into organisms. And we can take into this computer system that we built, called Sympatica, and that allows you to take this part list and take this basic chemical reactions, and you can sort of design these things. So you get these chemical reactions that get automatically translated into the corresponding equations with change of variables and all those things. And then you can, by simulation, by other means, you can start to understand what they do in terms of some sort of cryptic structure, model, but a model in a different sense. And then you can ask questions like, does lacti oscillate between 2 values, lacti low and lacti high, etc. And the system comes back and gives you an answer, true or false. If it is false, it gives you a counterexample. So the idea here is the biologist now draws these pictures that takes various databases and asks questions. Okay, can actually, what we do is something even more, where, for example, we could take yeast cells. There are 96 different yeast cells. One is wild type, and there are mutants and double mutants. And then you can ask the system to take a yeast cell cycle model and tell us a story. So you'll say this particular property is true about wild type, but it's not true about this mutant, and it's true about the double mutant. So the idea is that now biologists sort of start generating the stories and see if it makes sense. Right now it's a shaggy dog story. It tells you everything, much more than we want to know. But with some other inputs, it can be tuned into something that one can really use. So here's how you query the system and you look at various outputs. This is done in a system called VALIS. It's a bioinformatic environment. But VALIS works with public domain tools, but it's very easy to link it to MATLAB and simulate to other tools. And then you get all these other things for free. So you don't need to use Octave or BLAST or NumPy, which are the things that are linked right now. So right now it's linked to public domain, but anything else can do. I can say a little bit more about that. Okay, so now I know that I'm going to run out of time. So I had a choice between going more into the computational things or biology, and I decided to focus on the computational issues because it's a choice I made. It was the wrong choice. So there are various issues here. One of the simple things you may want to do are the simulation results, and there are a number of questions related to numerical analysis. Mostly, what are the good numerical integrators with algebraic constraints that can generate traces? Most people have not paid too much attention to these kind of questions. Another is we want to reason about these systems. So we want to build what are called cryptic structures. We'll come back and talk a lot more about that. So these are essentially some sort of finite automata that tells you different interesting states, qualitative states of biological systems, and then tells you how to go about those. And from that, it can engage in this query-answer sessions. The interesting thing is that if you just naively build the structures, the number of states will blow up fairly quickly, and the complexity is hard to control. But then you can do various things such as projection, but then you introduce non-determinism. And one way you handle that is by introducing invariance, so you end up with hybrid automata. So we'll talk about that a little bit. The questions of log reduction. So things that are by simulation equivalent. So I have a very complex model that is very detailed in biology. We've done collapsing and reductions. I want to make sure that the truth properties that I have are preserved. At least the truth properties that I can describe in a specific language of model logic, those are preserved. So that I never give the wrong answer to a biologist. I may constrain him from asking me harder and difficult questions. So expressive power of language could be constrained. But if he asks me a question, I should give the correct answer, independent of whatever model reductions I did. There's another question of symbolic computation. So here you want to avoid numerical integrators and do something that's very closely related to symbolic model checking. But here, because most of the underlying objects are algebraic, you want to do create Trotsky sentences that will encode your hybrid automata, and you want to reason about those. And then there are questions about various logic and model checking, and of course, all of this should be invisible to the biologist. You want biologists to interact with these systems through good visualization and natural language tools. Okay? So those are sort of the things that we want. So we want to handle a very large class of biological systems, and also we would like to be able to combine them. Some of the models could be qualitative, some of them could be detailed ODEs, but we should not be able to, we should not fail because part of the systems don't conform to our structures. So essentially what we have are a system of equations like this, has a set of dependent variables, set of independent variables, and some differential equations with algebraic constraints. And out of these, we can create these traces. The traces are temporal evolution of various chemicals, and our semantics is completely defined by these traces. So some initial value we can look at the discrete scale, because we know that already that we can't deal with differential algebra. So we are going to turn them into algebra, semi-algebraic geometry, by doing discrete sampling. And the idea is that this discrete sampling rate would be completely under control of the computer. So whatever you do with the numerical integrators, by Euler's can be encoded into this. I'll come back and talk about that. So we're going to define the semantics of our biological system in terms of these traces. You could have done that just by describing the flow. So the flow would describe that if I am in this state, and I wait for time s, what state would I end up at t0 plus s, or t0 plus 2s. Right. So as long as I have a little black box that says the local evolution for the flow, or how I go from one state to another in that discrete jump, I'll define my entire semantics based on that. Okay. And out of this, now that we have states and state transitions, and some initial state and some other final state, you could describe that in terms of this automata. That by itself is fairly simple. So far there's nothing new and difficult. So we can take the systems and write down this state. And once you have the states, you can reason about it. So one of the problems is that you would like to reduce the complexity of these 50 structures. So one thing you can do is projection. So that means, so I have a set of input variables, so set of state variables, set of output variables. I want to eliminate the state variables. So I want to get an input-output description. That I can do in community algebra or linear systems. But in differential algebra, we don't know how to do elimination of variables. There's no elimination theory. For the same reason, there's no decision procedure for membership. But we're going to do all of this in this sort of discretized model. So we'll do various projection, and also we may do collapsing, where we take long traces of temporal evolution and collapse into short transitions. That itself is problematic, because that will introduce non-determinism, because I would like my automata to have convergent property. That means if I go to a state, how I get out of the state should not depend on how I got in. So I would like that convergent property to be true. But if I have a hidden variable, that's going to control how I get out. And if I don't remember those hidden variables, I'm introduced non-determinism. And that's going to be, they'll immediately take away my bisimulation property. So one approach to get around that is that you don't need to remember all the hidden variables, but just enough so that you can tell how to resolve the non-determinism. And they can be encoded into flows and invariants. And you turn this whole thing into a hybrid automata with those flows and those invariants. And they will tell you how to resolve the non-determinism. So because of that, you have to get into hybrid automata. So that's sort of the story so far. And you get this sort of hybrid automata. And the nice thing is that the kind of moral logic, temporal logic that we want to use here, that remains decidable over this automata. So there's a decision procedure to do this. I was going to go more into this, but I think I'm going to run out of time. So these will be more precise than what you could obtain by just doing simulation with Euler method or any other approximations.
So can I ask a question?
Yeah.
So the biologists oftentimes will manage that the person that synthetic rock. I mean, for them to interact with this other tool, what sort of thing they may be looking for in the sense that they're looking for another possible network that
Right.
or with some additional components that they can use in order to modify the behavior of the rock.
Sure.
As a user.
Right. So first of all, the people who actually did those synthetic circuits are not biologists. They are ex-condensed matter physicists pretending to be biologists. Stan Libler, Elowitz, Gouet. So they are actually physicists with very strong quantitative background. And they taught themselves how to do this class maths. The other question is, why would they need a tool like this? So one of the things that I'm not going to talk about, but I actually wrote a perspective about that in science, was the question of designing combinatorial circuit. Not combinatorial circuit, combinatorially designed circuit. So the idea is that you take a set of genes and design all possible combination circuits and somehow find the ones that are interesting and then combinatorially connect them and find the next level of interesting things. And so one of the things they did, they took 3 genes, the same 3 genes that we talked about, and took 5 different promoters. And they could connect them in all possible choices to create 125 different circuits. And they could actually, it's a little bit tricky because the lag, E. coli already has a lag gene. So So the lag here and the color lag could interact. So there are two strains, one that has lag and the other one that was lag mutant. So one of the difficult parts was to actually figure out which circuits are meaningful and which circuits are trivial. I mean, the one circuit is nothing talks to anything that completely disconnected out of this 125, right? That's not an interesting circuit. So these kind of tools could be useful for them to design circuits. We're doing combinatorial all possible schemes and we check which ones of these have interesting properties that are bound to be interesting. But so I believe that those are the kinds of ways we can use introsimility biology. I'm going to skip all of this. So it's very interesting mathematics. But yes. Right. Right.
Physics, that is only left. And if you left it, you're going to choose environment that presentations, advertisements, loading, which is what I would call behavioral systems, implementation can behave realized in structure.
Right, that's a question that we are not addressing.
So for instance, go back to the again, you say, can I realize the behavior? Right.
So, right, that question is not being addressed.
Because that's what I would get the rules to come up with.
Well, here we're trying to go top down. So we have chemical reactions. The best place is that we're trying to understand biochemical reaction that already exists. So we're trying to create something and then realize influences. That will come up again because there are a lot of things we are not modeling. But even for the simple example of the biochemical circuit, the plasmid, that actually doesn't work the way I said. Because you could have a transcriptional factor bind to something, transcribe one gene. If it doesn't hit the stop codon properly, it could go a run-on and transcribe the next gene. And you do have those leakages. Another one is the TED-R protein. Once it's expressed, it will bind with other things. It will change the degradation properties. That's not... There's a lot of these physical things, how it diffuses within the cells, what happens in compartment. Even a simple thing like cell volume. The cell is growing. The concentrations are changing, but the volume is changing.
Can I do the version?
Yes.
It's even biological. Now, I have a biological system.
Right.
And then I want to do biological and biopreservation to figure out
If the property is valid. Yes.
the model is one out of five.
Right.
And I will start to say do that, something like that.
Right. So that we can do. Right. So basically you can describe all possible perturbations. And what it does right now is finds interesting properties that are true about one and false about when you perturb. If it is robust, actually I have an example on pure metabolism. I'll just strip it out because I have time. So I'm going to skip all of this, but this is sort of the mathematics. Just to come to, so these are the kinds of things people have done symbolic model checking, and these are the kinds of circuits we have tested earlier. And the interesting thing is that you can take the hybrid systems that you get. Right? So a lot of those logical interactions, in addition to the dynamics, continuous dynamics, can be written here. And one of the things, because of what I said, I can take these and model them symbolically. So for example, a formula that says eventually some property Px will hold true, right? I can write that symbolically, that there is a formula that tells me, algebraic formula, how I go from x to x prime. Could be a step of your integrator, could be a discrete, and Px prime is true. So that now I have quantified Tarski formula because all of these are algebraic, and I can do a decision procedure. But once you introduce, so there are many, I'm going to skip and just focus on one thing. One of the problems is that how do you define the next time operator? What does it mean to be next time? Because I have these discrete jumps, and also I have this continuous time, discretization that I have done. So, of course, I can have, and in order to do the symbolic model checking, I have to be able to write the least and the smallest, the largest fixed points in terms of these next time operators. So one of the key tricks is to come up with the good semantics for next time operators. And there is one that we have proposed. Both of them are semantically sound. One is you say in state S1 and you evolve, and at some time during the discrete interval, you jump to another state and you continue to evolve. So you have some discrete jumps, but you can have many such jumps. I mean, even with the first one, you could do xenopaths and other things. But this becomes tricky because this will come out as non-deterministic. But you could define the fixed points in terms of this. So the idea here is you're making lots of jumps in a very short time interval. And all of this can be translated into Tarski sentences. And then when you do quantified elimination, you have nice decision procedures. But also it turns out that there are certain convexity properties I can always write it down as positive null answers with one quantifier. But that means that I have much better, the basically have a single quantifier. And in some sense, I can get good approximate algorithms by mapping into a positive semi-definite program. So I could solve those examples in polynomials. Polynomial is the number of variables. So there are a lot of things one can do without sort of giving up because of the complexity issues. But I'm going to skip all of that. So actually this is already implemented, and one of the things you'll notice that, let me show you a one-state harmonic oscillator. One of the big problems would be these large numbers you'll get. Of course, with biological it doesn't make sense. But we are doing pure symbolic computation. So there are ways of avoiding it.
Large dynamic range, is that the issue here? The experience?
No, but the model actually is trying to reason exactly about the system. So instead, what you need are interval. So you don't need to do this, or you can just go into a finite field over large prime, then everything is controlled. So there are a lot of these kind of questions that still remain. So I'm going to skip all of this, and I'm already out of time. Let me give you one example, because let's go back to biology. So let's look at a particular problem of modeling apoptosis. And apoptosis is a process of programmed cell death. Please stop me whenever you want. I know I prepared too much. In this case, when a cell decides to commit apoptosis, it will shrink, the organelles will swell, the chromatids will condense, DNA will fragment, cell junction will get disintegrated, membranes will blab. That's an interesting word. Finally, will get engulfed. All of this will happen within 20 minutes. So cells will essentially explode. And of course, the DNA will, most of it will get cleaned up, the DNA will float around, God knows what happens to the DNA. Maybe they get incorporated by the other cells. So there's a theory of aging based on what happens to those lost DNA. Nature offers naked DNA. So if you put naked DNA, somebody will take it, right? Put them in the genome. Okay, so the theory here is based on these gas-phase molecules. And these are large molecules with a large subunit and a small subunit. There's a pro segment here. And at some point, it can get activated by cleavage. When that happens, this will turn around and go from an inactive state to active state. These casphases are going to determine when a cell has decided to commit suicide, and then it will go on. There's no stopping. Okay? Of course, cells can just decide this thing arbitrarily. They're not too finicky. Okay. So the picture one thinks of is that there's DNA. Suppose you have damage in your DNA, and at that point, you have cytochrome C released by the mitochondria. Right? The cytochrome C, and the cell already has abundant amount of alpha. Okay. And these things will dimerize to create this dimer. And a lot of the dimers will get activated, and they will make this molecule, large number of these large molecules that gets assembled fairly quickly. The moment after your DNA damage, the cell is trying to kill itself. At that point, there are these other molecules, caspase 9s, they will get bind to this coenzyme. These caspases will get activated by another caspase 3. And these large molecules then will go around chopping up all the DNA and proteins into pieces. And at that point, cell is dead. Okay? So this is the process. And there are still a lot of things that are unknown. So one way you can do is take the different kinds of cells, and by introducing RNAi, the RNA interference system, I can shut down FR1. So I have a cell that's going to express FR1, and a cell that does not express FR1, because that gene has been shut down. So I have all FR1 and no FR1, and I can mix them in different proportion. I'm trying to understand that biochemical pathway that I just described. There's this story that this is what happens with caspase.
The story is made out of pieces,
Right.
You have several
Reactions going on. Right, it's a cascade of reactions. Right.
And then rotation in
So I want to ask questions about how exactly apoptosis happens. If this story actually makes sense, like this, and the experiments actually show this. So this is looking at how I change FR1, and this is showing the concentration of caspases. So clearly the model doesn't explain the input-output behavior. So then the question becomes, this is the complete chemical pathway. I'm going to go into it. Why doesn't it explain? There are many ways it could. So for example, if I have written this as irreversible reaction, it's actually a reversible reaction. I could have the rates incorrect. I could be missing a reaction. This could be a nonlinear dual type coefficient, and I have the wrong dual type coefficient. Or there is this unknown molecule that binds here and competes with FR1. Okay, I don't know. It's all I know. This is the story in the literature, and it is not true. Okay.
So how do you even get the Hill coefficients in hand?
Some are from the literature. Some we didn't know, so we couldn't find the literature. We did a lot of experiments to compute this, but they are all done in vitro. So in vitro experiments. I'll show you why this project has taken so long. Because part of the problem is purifying each of one of those. And we saw how you get about one of the things we want to do is to purify the caspases and hollow enzymes, and that's actually a very tricky approach. So the one hypothesis is that when you have this F5 cytochrome BATP, this oligomer, and you are binding this caspase 9, most likely there's some nonlinearity that controls how the first one gets bound, or the second one gets bound, and then the kinetics are changed, so the cooperativity is changed. Okay. So here's an experiment we're currently doing where we've done actually, this works. But I'm not going to, we still have some unknowns. So we can take cell extract and basically take these molecules and purify them. So we have just the whole enzyme. So the idea here is to break up the chemical reactions into pieces and check each piece. We've already checked 2 pieces. There's a middle piece we still don't know. And we need to compute this Hill coefficient. So I was going to show you a demo of the system. Actually, this is used by biologists as they tinker with the experiments. They actually check each parameter, each value. So I'm not going to talk about this. I'm not going to talk about this for 2 minutes. Okay. And I'm going to stop. I'm not going to talk about it. So this is sort of interesting because we're also looking at spatial properties for C elegans gonad. I'm going to skip that. So there are some interesting models of stem cells and zones and aging. There's clearly no way to do it. So I'm going to start again with Book. So the question again comes back, was Book such a bad guy? Really, was he so stupid that he didn't see the reactive connection from theory and experiment? The answer is no. So I found this interesting quote where it says how theories and experiments should go hand in hand. So he says so many other things, of which the true philosophy depends. Again, we're coming back to truth. Of which, if any can reduce or weak, the whole chain is in danger of being dissolved. It is to begin with the hands and eyes, and to proceed through the memory, to be continued by the reason. Not is it to stop there, but to come about to the hands and eyes again, and so by a continual passage round from one faculty to another, it is to be maintained in life and strength. But yet, in his lifetime, he never got any respect. So when somebody actually did a play on him, where there was a character called Sir Nicholas Gimcrack, in a play called The Virtuoso by Thomas Shadwell, and when Book saw the play, he said, "Damn dogs, Hindi got my dues. God gave me revenge. He never got that. This is the last quote that is said to the Royal Society. And I have the misfortune, either not to be understood by some who have asserted I have done nothing, or to be misunderstood and misconstrued for what ends I now inquire not by others. And though many things I have first discovered could not find acceptance, yet I find there are not wanting some who pride themselves on arrogating of them for their own. But I let that pass for the present. So I think we'll have a better story than the book story. So.
Thanks a lot.
And that's not about time, but
First, I want to remind people that there is a roundtable discussion tomorrow at 10 o'clock in the A.B. Williams Building, room 2168. That is the ISR conference room. You're all welcome to come to that. But it's been asked many questions, and the thing that things that fast. And we'll try to fast. And any time for a question or two? Yeah.
No, we haven't done that. Mostly because we don't have funding to do it. It's actually very complex task. There is a group there are many groups. The one I know is in Mount Sinai School of Medicine under Ravi Iyengar. They have been doing these models of RAS pathway, Bhala and Iyengar. So these are patches of map-kinase and things like that. And they got funded by a P50 grant to take those things and put it in a database. I think it's called SIGPAD. So if you do a web search, okay, that right. It's the IEEE handbook for synthetic biology. That's a different part. So there are people in MIT. This is mostly from Knight and Drew Andy. They are supposedly creating such a spec for every transcriptional factor, every promoter sequence, every gene. I haven't seen it. So that would be an interesting thing to do. But these are, heroic tasks. 10 or 15 times magnitude of a human genome project, right? Right.
Because we are that.
Right. We don't, this is sort of a bizarre thing. Biologists want to understand this chemical pathways, but they are reluctant to measure the kinetic parameters. You don't get publications if you measure a kinetic parameter accurately. And yet you want to think about this very accurately. And there's even sort of discussions which indicate that many of these parameters are made up. People actually cheated. They cheated, they wrote papers, this is made up a parameter. But they wrote it as if they had experimental verified. Even if you do that, there's one other question, that's a good question. A lot of these parameters are computed by in vitro experiments. There are only 2 or 3 people who do it very accurately. Even if you get something right in vitro, they may not be correct in vivo. Because there are other effects. Because these are not a big bag of enzymes, things are not freely diffusing and freely interacting. They're being chaperoned, they're being transported in a very directed way. So one of the ways to do parameter searching, my approach would be to write down the reactions, write down the parameters as symbolic variables, take a cell, make many mutants and double mutants, and all of them will satisfy the qualitative properties only for one value of the symbolic parameter that you can search with these systems. So you just write, will come out as a solution to these big sets of polynomial equations. That's the correct in vivo parameter. Well, you can model that also dynamic changes to the parameters. Yeah, but in most cases, that could be written as a change in conformation. That could be part of my hybrid developer that says when I go from this mode to the other mode, my parameter may change. So I can have different parameters. But I could do, but I think that's the best way to find in vivo parameters.
I would like to take this discussion further tomorrow to the roundtable, but I want to thank everybody for coming, our speakers, for our